Buongiorno a tutti. Good morning to everybody. We are great honored and thrilled to have here Professor Kurt Wüthrich, which is a new fellow at the Accademia Lincei. So we are happy also to have here among us that is going to present a conference on DNA, RNA, protein history, place in perspective. Professor Wüthrich has done so many things that it would be very difficult to summarize in a short time, for example, on protein structure using NMR, and he received an incredible number of prizes. The Prix Louis, among them, I will only mention the Prix Louis Jeanneret Prize de Médecine, the Kyoto Prize in Advanced Technology, and the Nobel Prize in Chemistry. So I give the floor to Professor Wüthrich without losing any more time. Thank you. Professor.
All right, so we will see slides from a different lecture. But fortunately, I like pasta, and therefore I don't need my belt as a piece of clothes, and I can use my belt for the introduction. So when I talk about the molecules of life today, then I mean those molecules which are so-called biopolymers. And these are, you all have heard about messenger RNA because of the COVID-19 vaccines. And besides messenger RNA, we have DNA and we have proteins. And all three classes of biopolymers can be represented with my belt. They are linear arrays of chemical entities. In the nucleic acids, we have 4 building blocks. In proteins, we have 20 building blocks. And what is important, there is a beginning and an end which can unambiguously be distinguished, so that these linear arrays can be read off. Now, as far as DNA goes, we know that DNA is copied into a second belt to make messenger RNA, and then the sequence of nucleotides in the messenger RNA is translated into yet another linear array of amino acids. Now, as far as DNA is concerned, we have the famous DNA double helix from Watson and Crick in 1953. But that's enough for us to understand, we think, how DNA functions. In principle, you can read off the sequence of nucleotides and transfer it to the messenger RNA. However, that's not the whole story, because we know of only about 3% of the DNA in our genome what it is doing. And more than 90% of the DNA sequence are unknown, often referred to as non-functional DNA, but I cannot imagine that it is non-functional. I think that the future will bring us a lot of surprises with these so far not read parts of the DNA being involved in different control mechanisms in our metabolism. As far as messenger RNA goes, in principle, we can also understand a lot of its functioning by just looking at the linear sequence, because this linear sequence is read off and translated into proteins. When it comes to proteins, and now when it comes to proteins, then we can no longer in any way understand its wide range of different functions, which go from enzymatic activity to structural functions like hair or skin is protein. We have transport proteins such as hemoglobin, which has been famously studied here in Rome for many decades, and so on. And we cannot understand how a linear chain of amino acids can perform these widely different functions, and therefore we have to know about the three-dimensional arrangement of the polypeptide chain. For example, by forming a helix, like this. Okay? Now these considerations apply in part also to the messenger RNA, because today we know that many reactions are catalyzed by RNAs, not only messenger RNAs, there's also ribosomal RNA, and of course we suspect that there was an RNA world billions of years ago, which started today's kind of life that we know on our planet. Now what I will do today is to concentrate on discussing with you three-dimensional structures of proteins. And I will now follow the slides that I had prepared for a different lecture, and it will serve me well to pursue this subject. I'm currently working in this field in Shanghai at the Human Institute of Shanghai Tech University, and this is a remarkable picture because it shows my research group on March 29 of this year. 2 days later, these students were locked up in their bedrooms for 10 weeks. Lockdown on campus, 2 students per room, 10 weeks with visual control. They couldn't leave the rooms. Now they are quite happy again, except they must be tested 5 times a week, 5 PCR tests per week. And right now, Beijing is locked down. My associates' children who live in Beijing can not go to school at the moment, and so it is very difficult for us to work in China. What I show on this slide is our most recent study of a G-protein coupled receptor. G-protein coupled receptors are an abundant part of our proteome. They are membrane-standing proteins that transfer signals from extracellular compounds, for example, drugs, to the interior of the cell, where it induces a cascade of follow-up reactions that should lead, and do lead in many cases, to the desired effect of the drug. We have actually 826 different GPCRs in our body. And since 2007, it has become possible to determine three-dimensional structures of human GPCRs. And this is one example. This is a neurokinin 1 receptor. And we studied here the complex with a inhibitor, aprepitant. Now, aprepitant is an approved drug to fight nausea due to chemotherapy. And it has been in use for about 20 years. For us, it has a unique feature. in that you can see here, should this also work as a pointer? Or do you have a... No, I'm looking at this device here. Is this also a pointer? No, okay. Never mind. Can you go back? Return, well, that's up to me. Okay, the unique feature is in the center of this slide, where you see the chemical structure of this drug molecule. And at the bottom, you can see that it includes an aromatic ring, which is trifluoromethyl substituted in the 3 and 5 positions. And this aromatic ring, this substituted phenylalanine ring, is attached by a single bond to the remainder of the structure of the drug molecule. Now, on the left, you see an outline of the crystal structure of this receptor, and on the right, we have increased the detail at the top, that is the extracellular surface, and you can see the inhibitor, a prepitant, I mean this drug molecule, when it is bound into the orthosteric binding site of this GPCR. Now, the point to be made here, to let you understand what it's all about, when we dissolve this drug in a solvent, for example, DMSO, or in H2O, then the resonances of the two trifluoromethyl groups are identical, and we see a single line. When the drug is bound in the interior of the protein, then the two trifluoromethyl groups experience a different environment. We talk about a different microsusceptibility, which causes the two trifluoromethyl groups to give two different resonances that are about 2 parts per million. That's a unit that we use instead of frequency units to describe NMR spectra, P1 and P2. And within each one of these two states, we can distinguish 3 substates. Now, what this means is that the ring sits in the interior of the protein, like my hand, and we have one trifluoromethyl group where my thumb is and one at my little finger. They see a different environment. And in addition, the drug takes on 3 slightly different orientations. And now, when we look at this more closely, then we find that there is exchange. You can see this is a 2-dimensional 90 fluorine EXI experiment. EXI stands for exchange. And this, you see on the left, a contour plot of a 2-dimensional spectrum. And whenever we see cross peaks, so-called cross peaks, which are identified as C for cross peak, then this indicates to us that the ring is not just sitting in the protein, but that it flips over. And it flips over at the low frequency, in this case, like about 1 times per second. So this is a very slow dynamic. So we have this ring which sits, which fluctuates a little bit among 3 positions, and then it flips over. And in order for the ring to be, you see here we measure this quantitatively with saturation transfer experiments, which I do not want to describe in more detail. what is important here is that we inserted the GPCR into a nanodisk, that is a near natural environment of lipids, and in this near natural environment, as well as in detergent micelles, we can observe these low-frequency fluctuations. Now, what does it mean? It means that the binding site for the drug has to open about 8 angstroms. That's a tremendous change transient change in conformation. And, no, what's this? When we now compare this, this would be the red bar at the end, which would actually go through the screen. When we compare this with the 29 pairs of GPCRs, for which an active-like form, the complex of an antagonist, and with an agonist, and the inactive form, the complex with an antagonist, have been determined, then we see that the fluctuations that we see with these NMR experiments are about, I would say, 3 to 5 times larger than what has so far been known from crystallographic studies. And from this, we can also make sense of the observation that about 80%, 90% of the complexes of drug molecules with GPCRs do not provide high-resolution diffractions in crystal structure determinations. The explanation is that because of the large fluctuation, I'm now talking about the picture on the left, a large range of different compounds can be selected based only on size and shape, and it is only in the interior of the binding site that selection of active, of ligands with different efficacy is performed, and the crystallographers pick up many of these complexes with non-effective ligands, and therefore cannot obtain high-resolution crystallographic data. Overall, it is the typical today's use in my laboratory and most of those of the colleagues working with solution nuclear magnetic resonance to describe the dynamics of the molecules for which cryo-EM and X-ray crystallography have provided high-fidelity molecular architectures. And of course, knowing now that we have these large fluctuations of up to 8 angstroms in opening of the ligand binding site gives us an entirely new line of thinking in the field of design of drugs that target GPCRs. Now, I come back to the talk that I had actually planned here. You have now heard about our very latest research, which I had not originally planned to present here. What I wanted to demonstrate to you is that the development of methods that are used for studies of three-dimensional structures of biological macromolecules extends over 2 centuries, and that we are basing the procedures that we use on knowledge that has been obtained entirely by curiosity-driven research by individuals who had no idea it could be used in the ways we do, for example, because NMR had not been known at the time when it was done. Now, the first key observation regarding the use of the NMR technique for studies of proteins dates from 1827, when a botanist in England observed that pollen that he let fall into a beaker with water, and he was working with flowers, and then he was not careful, lost some pollen, and then he took the microscope and looked at this pollen, and to his surprise, this pollen moved around on the surface of the solution. And Mr. Brown published this observation without having an explanation, and the explanation came almost a century later when Albert Einstein, working in Bern, very near to where I was born, developed statistical mechanics that can explain on a detailed scale why Mr. Brown had observed these stochastic motions of particles suspended in a liquid. Here you see a picture of Robert Brown, and here a schematic of what he had seen under the microscope. When he had small parts of his plants, then they would move, translate, and change direction at high frequency. When he had larger particles, they would also change direction, but at lower frequency. And Albert Einstein then discovered, first of all, that these motions are due to collisions with the solvent molecules, which are at a high temperature. You see, the room temperature is very high temperature in physics, and so they have continuous collisions of the water molecules with suspended larger particles, and that gives rise to these stochastic movements. But Einstein published this explanation in 1905, and then with interactions with other physicists, it occurred to him that there would not only be translational motions, but that whenever these solvent molecules would hit not straight on to the sphere, but at an angle, then it would induce rotational stochastic motion. And these are the motions that have to be considered when we use NMR with macromolecules in solution. Now, you see, Einstein is known for his relativity theory, and he got a Nobel Prize for the magneto-optic effect, but he himself took his work on the Brownian motion as very important, and he would lecture about his explanation of the Brownian motion during the years after 1905, 1906. I find it quite amusing that Einstein was speaking in Bern to the Naturforschende Gesellschaft, and the attendance was 19 members plus himself. The meeting was held in a restaurant, and here you see in the abstract that he explains what he is talking about, and these are the translational and rotational stochastic motions of particles suspended in liquids. Another line of important observations before World War II were obtained in 1896 by Seemann, who, and I will show it to you in a minute, what Seemann discovered, the so-called Seemann splitting, and then we had a line of molecular beam experiments and optical pumping experiments that resemble NMR but do not represent an NMR experiment. And why is this so? The reason is that it is very difficult to observe transitions between the energy states that are relevant when one places nuclear spins with a spin number different from 0 into a magnetic field. These are very small energies and require sophisticated radio frequency equipment and radio frequency procedures, and these were developed during World War II by many thousand scientists in the USA alone who developed ever-improved radar to detect enemy planes. And as soon as those scientists went back to private life, many of them started to work in universities, and immediately after the war in 1946, they succeeded in directly observing transitions between the Zeeman states of nuclear spins in a magnetic field. And then we have here a whole line of developments from 1946 to 75, including the availability of NMR spectrometers in the US and in Europe, and with regard to biological applications, the developments of Fourier transform NMR and of two-dimensional NMR. You see here Felix Bloch. He is the first Swiss who got a Nobel Prize for NMR. We got exactly half of all prizes that were given for NMR went to the ETH. Bloch was a student of the ETH, and he shared the prize with Ed Purcell. And then we go to two-dimensional NMR, and that provided the second Nobel Prize for the ETH with Richard Ernst, with whom I had a very good and close collaboration over many years. Now, under what background was the NMR method of protein structure determination pursued? Because the environment provided by other experimental techniques was as important as the development of NMR spectroscopy. So in the 1950s, the key events were the development of, the model of the DNA double helix and the first crystal structures of proteins, hemoglobin and myoglobin. And here I showed to you a presentation of the hemoglobin molecule, which at that time was already studied intensively here in Rome, at the time when Perutz got the Nobel Prize for this achievement. There is no question of atomic resolution. The model is built with small pieces of laminated wood, 1/2 centimeter thick, which was pasted in three-dimensional shapes that followed the electron density maps. And I would say the official acknowledgement of modern structural biology happened in 1962 with the award of the Nobel Prize to Wilkins, Crick, and Watson for determining the DNA double helix, and to Perutz and Kendrew for the first crystal structures. I want to add that the DNA that was used to obtain the data on which DNA double helix was based was given to Professor Wilkins by my chemistry professor in Bern, Rudolf Siegner, and he is only mentioned on the side that he has really provided the material that enabled the development of the DNA double helix model. Today this would be handled very differently, because today we all know that the real bottleneck in structural biology is the preparation of the suitable samples for the experimental studies. In the 1960s, we have an important frame of reference that I don't want to go into in the first 5 years of the 1960s. A big step into modern biology was completed. And during these times, the first NMR experiments, in the 1960s, the first NMR experiments with proteins in solution were performed. And the key observation in these early endeavors was to show that one had to study the intact proteins. There was no way around studying the complexity of the folded polypeptide chain. You see, you could imagine that you would, for simplicity, cut the polypeptide apart, study individual pieces of it, and then generate the structure. This was already in the 1960s, was shown to be impossible. When you unfold the protein, you get the spectrum at the bottom. When it is folded, you get a much more complex spectrum of the folded protein. So it was clear one of the elements needed to ever get a structure of a protein by NMR had to be experiments done with the folded protein. You have here a sketch of why this is so. In the RC form, that stands for random coil on the left, all parts of the polypeptide chain are in the same environment, are accessible to the solvent, and you have here the same situation as I had explained with the drug aprepitant, where the two trifluoromethyl groups were equivalent in a solvent, in an isotropic solvent, but were split up when inserted into a protein, and exactly the same happens here, also in the absence of fluorine atoms for the resonances of the protons. Now, at the end of the 1960s, and that is 12 years after the first crystal structure of myoglobin was presented, we had only 9 different proteins for which the structures were known from crystallography. And these were myoglobin, hemoglobin, cytochrome C, and then a line of enzymes. Now, why is this so? The explanation is very simple. These were the only proteins that could be isolated from natural sources in sufficient quantities to undergo structural biology experiments. And because this was so, we had no other way with NMR than to use the same proteins, because there were no other proteins available in sufficient quantity. So myself, I have another point. You see, these 9 structures were presented in a book in 1969, a book by a crystallographer, Richard Dickerson, and an artist, Irving Geis. You see, there were no computers that were able to make drawings of a molecule. That came 2 decades later. And so the only way to present the results of a structure determination to a wider group of biochemists and molecular biologists was to have an artist make drawings. And this, for example, is a drawing of Irving Geis. That's on the cover of this book. It's a drawing of hemoglobin, and now you can see much more clearly the arrangement of 4 polypeptide chains and 4 heme groups in this molecule. You remember the pile of wooden platelets that we have seen earlier. And now one gets an intuitive feeling of understanding what this molecule looks like. Now, what Perutz saw was that when a ligand was bound to the heme iron, these are the red plates here in the center of each chain, then the distance between 2 heme groups changed by 12 angstrom. And the diameter of the whole molecule is about 50 angstrom, so this is a huge change in conformation. But what Max Perutz couldn't see at the resolution attained was whether or not there were also changes near the heme group in the hemoglobin. And so that's when I started to do NMR with proteins, and very first thing that I actually did in the field was to study my own hemoglobin. You see, it's published as hemoglobin KW, and what was observed is that there are lines. You see at the bottom of the slide, you have a scale which says 13 to 23 and -1 to -5. Now, normally you don't see any lines in this position. What these are all lines of the heme groups and of amino acid residues next to the heme groups. And as soon as we would change the ligation state of the hemoglobin, then these lines would move around, showing very clearly that there were also changes as well in the electronic structure of the heme group and the conformation, the tertiary conformation within the subunits. Even more striking is this example. It's also from the book of Dickerson and Geis. We are again at the experimental model with these wooden platelets pasted together, and then Irving Geis made an atomic resolution rendering of the structure, and interestingly, there is no 6 ligand to the heme iron. The reason for this is that the NMR spectrum had clearly shown that Dickerson's crystal structure had a mistake. He had a lysine residue bound to the heme iron, and from the fact that we have these lines, you have to look at the spectrum from -1 to -4 parts per million in the right lower angle, and this is a spectrum of methionine. And so at that point, when it was so clear that we could correct an experimental crystal structure, I started to believe that we could do 3-dimensional structures of proteins with NMR. In the 1980s, we had some very crucial developments again for our work. One is the sequencing of DNA and the polymerase chain reaction, PCR. And they both changed the practice of molecular biology and biochemistry completely. The laboratories were not the same after DNA sequencing became known, and they changed again dramatically when PCR was introduced. And now crystallography and NMR At this point, there were only these two methods. All of a sudden had access to a large array of different proteins that could be derived without going at the natural sources and just going at the DNA and express those proteins, for example, in E. coli or in other bacteria. And this then led to the development of the method of protein structure determination in solution. And this was not so straightforward. And I tried to explain to you why this is. You see, in crystallography, and more recently in cryo-EM, when you determine a three-dimensional structure of a protein, then you make an image, like photographic image. And for this, the molecules are isolated in crystals, and they are frozen down, typically to liquid nitrogen temperature, so that they are not only held still in a crystal lattice, but the thermal motions are greatly reduced by the low temperature. So you can take a picture. Now, the Brownian motions of protein molecules of the sizes that we can study with NMR, that is, well, it's about up to up to 9, well, I should be exact, up to 900 kilodalton. So this is pretty big. And for this, the frequencies of the Brownian motion were about 10 to the 9. I mean, the range from 10 to the 8 to 10 to the 10 per second. And these are stochastic motions, so there is no way you could take a photograph. And it took us many years to quiet down the crystallography colleagues about the fact that we could get sharp pictures in spite of these high-frequency stochastic motions. And it actually started in 84, and it was only resolved with a paper in Journal Molecular Biology, when Robert Huber and I had decided to solve the same unknown structure by Jedebar crystallography. We were much faster than he was. He didn't even have crystals, and he solved the structure, and there was the same. And then there was a 70-page article in JMB, and a comment by Sidney Brenner that things were now settled and they could be done. How is it done? Well, the idea is you have to find a parameter that defines a three-dimensional structure and is invariant under rotation and translation. That is, you have to have a scalar quantity that defines the three-dimensional structure. And the distance between atoms is this kind of a parameter. You can see in this drawing that the distance is invariant of the orientation of these handles. You see, the circles are atoms, and the lines are the distances between the atoms. And these are the same in the upper left and the lower right corner, and they are also independent of the orientation of these little structures here. And by 1982, we also, in collaboration with Professor Ernst, we had the necessary two-dimensional NMR experiments that enabled to solve the problem. I again use my belt. When you find a short distance between two atoms along the belt, then you define a closed ring. When you have two distances, you get already a relatively complex structure. And when you have a line of short distances distances that are far too long to be seen by our technique. And then we get a helix, and now these distances are short enough to be seen. Okay, and this is what the computer does for us. But for this, we had to be able to identify the positions along the linear chain between which we would see short distances. And that's shown schematically on this slide. Now, when we get such results, and when we do not get a very big loop, but when we start to get small loops, then we not only have the measured distance, but we induce a lot of collisions between the atoms. So these are lower limits on the distance. So by measuring a few hundred upper distance constraints, that is, constraints that produce such a closed ring, then we generate thousands of distance constraints, most of them being repulsive distance constraints because of close approach of the atoms under the influence of the experimentally imposed ring structure. And then we use mathematical tools, distance geometry, to go back from this n,000 dimensional distance space to the three-dimensional space of molecules. The first protein was BUSI. This shows you a two-dimensional spectrum of a specific kind. We call this NOSI. It shows nuclear Overhauser effects. Each point in this plane, this is now a contour plot, each point in this plane indicates one short distance. Again, it indicates one such loop. And so we have, even on this slide alone, about 200 such loops, and then the computer calculates a three-dimensional structure from this. I finished the talk here by coming back to what I described at the beginning. See, by now, about 15,000 protein structures have been solved by NMR, and about 2,000 structures of DNA fragments and RNA preparations. Today, the main interest in the technique is no longer in structure determination. It is very clearly in providing dynamic information on structures for which the geometry has been determined by either X-ray crystallography or cryo-electron microscopy. Here I show this with these different colors. This drawing is the first protein structure that we had solved by NMR, but in addition to getting the geometry of the molecule, we get all these color patches which tell us about dynamics, about intramolecular motions and rate processes, about interactions with ligands, with the solvent, or with ligands that are dissolved in the solvent. Most interesting in practice are here drug molecules or potential lead compounds in the science of drug development. And this is how the circle ends. We could measure such dynamic features of proteins already in the early 1970s. I mean, we had discovered ring flips of phenylalanine and tyrosine already in 1974. But we were not really accepted until we showed that we could determine de novo structures in solution, and now we can go back and concentrate on pursuing what is real strength of the NMR, namely to add information on dynamics and intermolecular interactions to the molecular architectures that can be determined either by crystallography, cryo-EM, or by NMR. And today it is particularly important to stress that in light of AlphaFold and other artificial intelligence approaches, which are now able to predict three-dimensional structures of a very large array of proteins, they greatly help the experimental determination of crystal structures and cryo-EM structures. It is questionable to which extent they can replace these techniques in the future by further refinement of the structure prediction methods, but they are worlds apart from being able to describe an intrinsically unfolded polypeptide chain in its functional state, or they fail whenever there are unfolded, flexibly disordered parts in protein molecules. And from this, I foresee that within the next 10 years, the importance of using these techniques for studies of these not rigidly structured parts of protein molecules, as well as of RNA molecules, will actually increase in importance. Thank you for your attention, and I apologize again for the mix-up with the slides, which must be entirely my fault. I do not know why we couldn't open the desk that was meant to be shown here. It was very similar, by the way. Thank you.
Thank you very much for this wonderful lecture. I think that there are some questions. Someone would like to ask something? Yes, Maurizio. Il microfono.
You just mentioned at the very end this fact that there are portions of proteins, generally domains, that are intrinsically disordered under native conditions. You just said it now. And now I have two questions. The first, some people claim that even in the cell, there is a substantial fraction of the proteins which are intrinsically disordered. And this is the first question, is what sort of fraction do you think sounds reasonable or believable? That's the first question. The second is, can you make a simple explanation to us that what is the function, why so many proteins are partially unfolded in the cell? See, if they are, I mean, there must be a functional motivation to keep them during evolution, because otherwise they are generally more, intrinsically more attackable by proteolytic enzymes and whatever. Sorry, it's a long question.
Well, I mean, when I look back at our own work, then the prion protein has a flexibly disordered tail of 110 residues. It is suggestive that this flexibly disordered tail gets entangled on the way to the change from the cellular prion protein to the disease-related aggregated prion protein. We have seen we have seen disordered tails in the homeodomain, for example. And when the homeodomain binds to the DNA, then these flexibly disordered chain ends get located snugly into the minor groove of the DNA. If they were structured initially, they would have to be unfolded in order to undergo this interaction with the DNA. So it is sort of indicative that having these tails in a flexibly, so to say, in a ready form for the interaction with the DNA has an advantage in the physiological functioning. And then I think the third answer to your question would be that it is unlikely that in the living cell these intrinsically unfolded proteins are not in some way aggregated with carriers. That could be lipids, that could be the surface of the cell membrane, that could be larger proteins, that could be RNA, and it is, I mean, you see, these intrinsically unfolded proteins are studied in aqueous solution, usually, or in salt solution. And that is not a sufficiently realistic representation of the high density of larger particles that surround these polypeptide chains in the cell. So here I would just say it is unlikely that in the physiological state these are really, these are per se extended, disordered, but attached to some scaffold that would impose a given structure on them.
Okay. Other questions? Sì. Lì in fondo poi.
Okay, thank you for your talk. Do you think that solid-state NMR can help shedding light on the liquid phase separation of proteins into the so-called membraneless organelles?
Well, this is certainly a very exciting prospect. Not only for solid-state NMR, the combination of solution NMR, optical methods, and solid-state NMR could bring the desired solution there.
Okay, thank you.
Just a curiosity. You had list of names with a star. Does a star mean a Nobel laureate?
Yes.
So my semi-serious question is the following. The first, in one slide that you show, the one with the names of Pauling, Crick, Rosalind Franklin, the only people without a star were either women or non-white people. Do you think that this is just a coincidence, or is it a sign of times?
Well, that's how history has gone. I mean, science was pursued starting in Western Europe and England. I mean, what we today consider natural science research, and it was essentially almost exclusively pursued by men. That is just a numbers game. You have very few women scientists who got the Nobel Prize before, in the last century. And that the reason is a numbers game. It's clear. If you don't encourage young women to pursue a career in science, you will not get female Nobel laureates.
My question was the names of women and Indians were there, but were the only ones without a star.
Well, Rosalind Franklin died before the Nobel Prize was given. That's reason the Nobel Prize is not given posthumous. So instead of her, the prize was shared by her boss. And they could have been my chemistry professor in Bern instead of her boss, because he gave the DNA that Rosalind Franklin, he prepared the DNA that Rosalind Franklin was measuring.
Okay.
In this case, it's clear. Did you see other names of?
Okay.
Yes, but here the answer is clear, I think.
Well, I'm sorry, but we have to move to next lectures, so we thank again Professor Wüthrich.